Hi students, now let us talk about the second part of the semi-conservative replication of DNA. Once again, look, listen and learn. In this module, I'll be talking about the mechanism of DNA replication. Now, replication mechanism basically involves several different steps. Here, I'll talk about the first step, and that is called as activation of nucleotides. Now, what is a nucleotide? You have learned about that in the previous modules. Now, we know that a nucleotide is made up of three components. Is that right? Here is the first one. What do you see? What you see here basically is a deoxyribose sugar. Is that correct? Then you know that as the first carbon of this sugar is attached a nitrogen base. What nitrogen base is this? It says A, which means it has to be adenine. Is that correct? Right? Then on the fifth carbon of the same sugar, I'm going to see a phosphate group, right? Yes? So together these three things will make up a nucleotide. Now I'm going to give it a name. Because it has adenine as a nitrogen base, I'm going to call this molecule as DAMP, which stands for, it's a heavy word, look carefully, deoxyribo adenosine monophosphate, because it contains only one phosphate group. Is that okay? Now these monophosphates are seen freely circulating in the nucleoplasm. Now my step here is to activate these nucleotides. Now remember, to activate anything you require energy. And we also know that the energy for a living cell is going to be ATP. So even for activating nucleotides, we require ATP molecule. Now to complete the reaction, we require an enzyme. And the enzyme that plays a role here is called as phosphorylase enzyme. So for activation, we require two things. One is ATP, the other is phosphorylase. Is that okay? Now what happens during activation? Let us look. Okay, I'm repeating the same molecule of DAMP. Now I'm going to add one phosphate group. Look, a phosphate group just got added. Now I'm going to add one more phosphate group. See what happened? So I did what? I just converted an AMP to an ATP, and I'm going to call this molecule as DATP, which stands for deoxyribose adenosine triphosphate, because it now has two more phosphate groups. Is that okay? And what remains in the end is a molecule of AMP. So the overall reaction for activation will be DAMP along with a molecule of ATP in the presence of phosphorylase enzyme will give you DATP and a molecule of AMP. Is that okay? And that is how you activate a nucleotide. Now remember, DNA molecule has got three more types of nucleotides based on the different nitrogen bases. Let us go and activate them also in a similar fashion. Now what do you see here? You basically see here another nucleotide. This time it has another nitrogen base. And can you tell me what the nitrogen base is? You are absolutely right. This one is guanine, right? So if the guanine is a nitrogen base, I'm going to call this molecule as DGMP, which stands for deoxyribo guanosine monophosphate, because it has got only one phosphate group. And how do we activate this? Once again, we use phosphorylase enzyme and a molecule of ATP for activation. And what do I get? I get a compound which has got now three phosphates attached, and I'm going to call this compound as DGTP, which stands for deoxyribo guanosine triphosphate. And you'll release a molecule of AMP. Now let us look at another nucleotide, and look at this picture right here. What do you see? Another nucleotide. And what nitrogen base do you see here? You guessed it correct. It is cytosine, right? So if cytosine is my nitrogen base, I'm going to call this molecule as DCMP, which stands for deoxyribo cytidine monophosphate. Once again, I will activate it in the presence of phosphorylase enzyme and ATP molecule. And what do I get? I'm going to get a triphosphate. What will I call this triphosphate? You guessed it right. I will call it as DCTP, which stands for deoxyribocytidine triphosphate. And I will liberate a molecule of AMP. Last but not least, we have another nucleotide. Which nitrogen base does it have? You guessed it correct. It has a thymine. Now, if thymine is a nitrogen base, the nucleotide can be called as DTMP. You guessed it correct one more time. And the name for this will be deoxyribothymidine monophosphate. Let me go ahead and activate it in a similar fashion using phosphorylase and ATP. And what do I get? I get a triphosphate here. And what will I call it? I'm going to call it as DTTP, which stands for deoxyribothymidine triphosphate. Is that correct? And I get a molecule of AMP. So basically, this is how you would activate your nucleotides using phosphorylase enzyme and molecule of ATP, and you will convert the monophosphates into their triphosphates in the nucleoplasm. Is that correct? And this brings us to the end of this module, which is part 2 of the semi-conservative replication of DNA. In the next module, I will be talking about the third part of the semi-conservative replication of DNA. Thank you and take care.